trouble seemed so far away. Now I'd look.
Endomembrane trafficking is described as the movement of material among endomembrane organelles. In addition, it is vital for transportation of proteins and macromolecules to certain destinations in the inner and outer portion of the cell. The starting point of this process begins in the nucleus, which is classified as the control center. The nucleus is surrounded by a double membrane and contains DNA, deoxyribonucleic acid, RNA, ribonucleic acid, and proteins. Located inside of the nucleus is DNA. DNA is the carrier of genetic information and stores instructions for the formation of proteins. The procedure of manufacturing proteins is classified as protein synthesis. Protein synthesis encompasses transcription of DNA and translation of RNA. DNA, deoxyribonucleic acid, is formed by two DNA strands. The two DNA strands are connected by hydrogen bonds that are detained by nitrogenous bases found in nucleotides. The four nitrogenous bases are adenine, thymine, guanine, and cytosine. Adenine pairs with thymine, and guanine pairs with cytosine. mRNA, messenger ribonucleic acid, is the RNA fragment that transports chromosomal information from the DNA, deoxyribonucleic acid, to the ribosomes. This process is described as transcription. Transcription is the indocitrination of genetic instructions on a component of messenger ribonucleic acid. In other words, this process concludes of copying instructions from DNA, deoxyribonucleic acid, to mRNA, messenger ribonucleic acid, in the nucleus. In transcription, DNA is unzipped, and the enzyme RNA polymorph goes through the template strand of the DNA. As the RNA polymorph goes to the template strand, the corresponding RNA nucleotides will be added to the DNA nucleotides. When the single helix mRNA strand is complete, the mRNA strand will separate from the DNA, and the DNA will rezip into a double helix. This process forms RNA. RNA contains the same nucleogenous bases as DNA, except thymine is replaced with uracil and RNA. The complete messenger ribonucleic acid contains a categorization of three nucleotides known as codons. The group of three nucleotides will code for a precise amino acid in translation. mRNA transports from the nucleus through nuclear pore to a ribosome in the cytoplasm enclosed by amino acids. This process is classified as translation. In this procedure, ribosomes create proteins. mRNA is translated by a ribosome to create a precise polypeptide chain of amino acids. tRNA has two attachment sites. The first is an anticodon, a triplet of nucleotides that base pairs with an mRNA codon. The other attachment site binds to an amino acid, the one specified by a codon. Translation begins with the binding of the ribosomal subunit to a specific sequence on the mRNA chain. Ribosome subunits and a tRNA converge on an mRNA. A tRNA binds to the codon, forming a peptide bond between the amino acids. The tRNA is released, and the ribosomes move to the next codon. Post-transition modification include excision of the polypeptide chains. This procedure involves the polypeptide chain to move into the endoplasmic reticulum, where certain substances are added to the polypeptide, such as sugar and protein salts. Next, the protein is carried by a vesicle to the sysphase of the Golgi apparatus, causing the Golgi to develop to a medial Golgi. Then the protein is carried to the transphase and transferred to the plasma membrane. The excessive code is cleaned up by a lysosome. The proteins then may possibly be transported to the outer portion of the cell through a secretory vesicle. By exocytosis, a vesicle transports to the cell surface, and the protein-studded lipid bilayer of its membrane combined with the plasma membrane. As the vesicle is deprived of its identity, its contents are released to the environment.
I'm not half the man I used to be.